You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri, and I'm your host, Dr. Jay Portnoy. It consists of a series of presentations by experts in allergy immunology and can serve as the didactic curriculum for trainees in the field. I hope you enjoy COLA.
I'll let you just get started then. Thanks.
Okay, we're going to talk about aerobiology today, and the part of aerobiology that allergists are really interested in is air sampling for pollen and spores, because these are the main triggers that you find in the air that trigger the asthma that you need to treat, diagnose and treat the way we saw in the previous talk. So my disclaimer is that I'm retired, and other than a very small amount of consulting for Clorox Corporation that I did a couple of years ago, I only get paid by the U.S. government and anything I've saved over the years. So our questions that we're going to address, and this kind of comes from the old way presentations were made. You used to always have to have questions, but I thought it was a good thing to let people concentrate on what we're actually talking about. We want to look at things that are used to collect air samples. So which of the following is commonly used to collect air samples for aeroallergen estimation? And this really is a statistical estimate that we're making. We call it a pollen count, but it's actually a pollen statistical estimation. You can use a weather balloon or a remote control aircraft, or a house dust or a Hurst spore trap, or a spectrophotometer. And then which of the following is a typical aeroallergen in Kansas City? Oak pollen, ragweed pollen, elm pollen, grass pollen, or all of the above? And then which of the following mathematical manipulations is used to produce airborne pollen counts in pollen grains per cubic meter of air? Which is a question that people think is really hard, but is actually extremely simple, and it's just the answer is described in the question. Okay, we want to understand how air samples are taken for outdoor pollen and spore counts. We want to understand a little bit about indoor spore counts and why we might do indoor spore counts. And we want to review the major outdoor and indoor aeroallergens in the Kansas City area. And this is a short list. There's certainly a much longer list, but these are the major ones, 90+% of what's in the outdoor and indoor air. So the overall name for the field is aerobiology, and aerobiology, as it describes, is anything that's biological that's found in the air. And aerobiology is of interest to allergists because they like to know what's in there. They like to know how it correlates with their patients coming into the clinic. And also you find that a lot of allergists are actually sort of amateur botanists, and they are really interested in what's growing outside, and they can go out in the field and identify the first dozen or so plants they come to, and know the allergens that come to them, and watch the seasons and know what time things are blooming, and what's blooming when, and what to expect to see at various times of the year. But aerobiology also is a big field in the study of biology, and even NASA has an aerobiology laboratory, and they say the answers to life are up in the air on their website. And so the atmosphere is home to a wide diversity of microorganisms, including bacteria. So you can find bacteria in the air. You can certainly find viruses in the air, as we've learned on our last 2 or 3 years of pandemic. I remember going to meetings at the CDC, and I was there to talk about pollen counts, but the argument would always arise as to how much airborne, how much transmission of virus was by airborne and how much was by fomite. Fomite is just, somebody who has the disease touches an object, and then you touch the object, and then you get the disease by touching your nose or something. And I always lost that argument that there had to be significant airborne viral transport. And I always lost that argument when I was at the CDC. And so it was really interesting to see them come around to the fact that, well, maybe some viruses are mostly airborne transport. Another thing that's airborne transport, of course, is fungal spores. And by studying these, we can learn answers to a lot of bigger questions. So aerobiologists study the organisms and particles of biological origin, bioaerosols, that float around in the planet's atmosphere. And so even somebody who's studying the migration of corn worms up from Mexico every year, as the corn crop matures, it starts to mature in Central America, and then it moves northward through Mexico and on into Texas, and then up through the United States. There's a cabbage whelper type insect that comes along with it, and it flies through the air in large swarms large enough that you can actually see them on radar. And so anything that's a biologic that's found in the air is aerobiology, but we're mostly interested in pollen and spores. And so that's kind of where we're going to stay today. There are lots of collectors that you can use to collect stuff in the air. Most of this is the height of 1950s technology. These collectors first started when we were really interested in radioactive particles that were being released into the air by all the atomic bomb tests that were going on in the 40s and 50s. And so people started to put out collectors for these radioactive particles, and then they discovered, well, you can actually find a lot of pollen and spores on these. And so we have collectors like the upper right-hand side, which is a typical Burkhardt collector. The middle slide here is the pollen sense collector, and I just added this picture to the slide set because this is what you have at Children's Mercy. This basically runs automatically. The tape cartridge in it needs to be changed periodically, and they do break down periodically, but basically this collects pollen and spore data on a minute by minute basis, and using the internet reports those pollen and spore counts to a central database, and it's using artificial intelligence to learn basically what's out there and sort of teach itself constantly and improve the counts that it produces on a constant basis. But eventually, I'm sure this company, and I went to their website and noticed that they're starting to build this. Eventually, this company is going to be putting out on minute by minute almost pollen counts, and they're also going to be putting out 2 or 3 day forecasts. So they're going to be a good source to see what the pollen count's likely to be in the next hour or so, in the next day or so. And that's the collector in the middle there, the pollen sense collector. Then right down below it is an interesting technology that basically doesn't pull in air. It uses a spinning rod. This down here at the bottom is a lucite rod sticking straight down, and the lucite rod is coated in grease, and it spins around and bumps into pollen, and the pollen sticks to it. And you look at that under the microscope and you figure out how many pollen grains stuck to the rod and produce a statistical estimate of pollen. And then there's an air sampler that's mostly designed to be an indoor air sampler, but we used it many times outdoors. And that's the old Allergenco air sampler. And there are newer versions of this, but all of these things are basically Hurst spore trap type samplers. And this was a publication from the 1970s describing an early Hurst spore trap. And basically, it's just a chamber that you pull a vacuum in that has a slide. In this case, it's really rudimentary. The slide is on a string. The string is wound around a clock that turns continuously, and the string is slowly pulled by a slit in the chamber. And so air comes rushing through here through the slit, and anything that's heavier than the air, like a pollen or a mold spore, goes smacking into the slide. The slide is coated with something like silicone grease or Vaseline, and it sticks. Anything that's heavy goes smacking into it and sticks. This is collected, in this case, probably over a week's period of time. And then you take the slide out and you stain it, and you look at it under the microscope, and you actually do an estimated count based on the volume of air you've collected and the number of pollen grains that have hit the slide. And so the Hurst spore trap has sort of evolved into the most commonly used collecting device, and this is a Burkhardt device. And for your information, Mr. Burkhardt or Mr. Willie actually in England, who owns the Burkhardt manufacturing facility, is interested in retiring, and the kids are evidently not interested in continuing the business. So I think Burkhardt is currently for sale. Anybody who wants to really get in the aerobiology business could probably buy his whole manufacturing plant, the ability to make these things. Unfortunately, these things last a very long time, and you don't sell a whole lot of them in a year. But this is the inlet here behind the square that's outlined over the face of the collector. There's a vane on here that keeps this collector pointing into the wind. There's a spinning Venturi in the chamber. Air comes rushing in through this slit and impacts a slide that's mounted on a collecting head that goes into the top here. And you can see this would be a day's collection, and this was probably collected by James Anderson in Ontario, and he most likely starts his collection about 7 o'clock in the morning or so. And so you can see this would be morning rush hour, noon, afternoon rush hour, and then this longer period would be overnight. So you can see there's a lot of stuff in the air that's not just pollen and mold. There's a lot of dust in the air. There's a lot of dirt in the air. As we get wildfires out west, there's a lot of smoke particles in the air. So there's a lot of stuff in the air besides just the pollen and mold. But the pollen and mold is what causes most of the inflammation. We could talk about pollution another day. This is how the sampler head fits on. It just basically, there's a spring-loaded rod on top that holds the sample head down. This is a close-up view of the inlet. Air comes rushing in the inlet, rushing right into the middle here where the slide is mounted. It comes smacking into the slide, or a 7-day device. And this is the same clock for both. And so you have to wind, there's a clock in the middle here. And so you have to wind this clock, which actually was probably the thing that gave us the most trouble about this device, was actually learning how to wind the clock, since so few people these days actually wind a clock. And this is how the slide is mounted in the 1-day slide collector. The collector starts at the top, moves down over the face of moves down behind the inlet, and you get a daily collection. And then you take this back to the lab and you put some stain on it, and you put a cover slip on it, and you look at it with a microscope. And knowing the area that you count, how many pollen grains are in the area you count, and knowing what your sample, your inlet velocity was, or basically how much air you pulled in to get that number of pollen, you can just simply divide the number of pollen grains per day by the amount of air you collected per day, and you get a pollen estimate or count that is essentially an average for the 24-hour period. So it could be higher, it could be lower, but the pollen count you get is a daily 24-hour average. And there, you know, there are fine points of this. It's marked where you need, if you want to start the 7-day head, it's marked where you start it. You just basically tighten down the nut. The air starts coming in here, and the little green dot is to tell you which way you're going, because if you take 7 of these off at a time, you have to pay attention and not get them on there backwards. Now, there are lots of other air collectors. This is a man from Cornell who used to liked model planes, and he had a grant to collect spores over tobacco fields, because there's a tobacco fungus that moves up from Cuba every year. As tobacco matures, it starts in Cuba and travels on the air currents and moves north in North America. And when tobacco was a major cash crop, they were very interested in finding out and knowing when this so he had an airplane with 4 petri dishes basically mounted on the front, and he had a little device on the petri dish that would cover it, and then upon command it would turn around and open up so that the spores would go smacking into the petri dish. And so he would fly this airplane at a certain height over the field, open the petri dish for 10 minutes, he'd close it, he'd move it to another height, he'd open the petri dish for another 10 minutes, and he'd collect at the next height. And you can see this was big doings, especially in North Carolina, because he's got the highway patrol out there and he's got the road blocked off, so he can play with his model airplane out there. But using these methods, a lot was learned about spores and pollen and the way they travel around long distances. Another device out there is a rotorod device. That was the device that was in the bottom middle. And the rotorod is now owned by by Surveillance Data International, and the name changes a lot as different corporations have bought this. But basically, they own pollen.com, and they have a facility in Plymouth Meeting, Pennsylvania, that collects data on lots of things. And they have a website where they sell information and they sell advertising, and they do an estimated pollen count for the US. In Canada, there's a very similar organization called Aerobiology Research Laboratories. And so if you're dealing with the rotorod devices, these two organizations sell parts and sell rotorod type devices, and I'll show you another picture of the rotorod devices in a few minutes. And they also produce pollen counts. They have websites, and they sell advertising on the website, and that's what and they sell data to drug companies who are producing allergy medicines and things like that. So there is some big business associated with allergy. This is pollen.com. They do a forecast report for anywhere you are in the nation, and sometimes their data is good and sometimes their data is not so good. Basically, pollen counts are strongly dependent on weather. They're strongly dependent on the temperature at any, how hot it's been for the last week or so, on whether you've had rain for the day, and whether you've got a cold front in, and whether you've got a lot of wind. So if the weather's normal, their pollen counts are pretty good. If the weather's abnormal, their pollen counts are generally not so good, because basically they're just, they've got some algorithm that averages out previous data and provides an estimated pollen count for the day. Now, the National Allergy Bureau is part of the American Academy of Allergy, Asthma, and Immunology that all of you are members of. And they produce a pollen count daily that's basically what the pollen level was at any of about 80 to 100 sites around the United States and Canada. And they actually have some sites in South America. So this is an actual pollen count of what it was yesterday. And what it was yesterday is actually pretty close to what it was today, and using the information of what it was yesterday versus the weather, you can come up with a pretty good estimation of what it's going to be like, what the pollen levels are going to be like today. And they don't turn out numbers; they turn out dashboard. They're actually using a dashboard scenario now, and they're doing it in 4 different categories: trees, weeds, grass, and mold. This is probably fairly early in the year, where the trees are moderate, but there are no weeds or grass to speak of. There's no weed or grass to speak of yet. And the mold varies more depending upon rainfall and depending upon anything else. Time of year, except in the dead of winter, you can always find a few spores in the air. This is a rotorod device. These are old prices probably, but they're the last ones I could find. You can see it's just basically a spinning rod that's got a piece of lucite attached to it. And lucite is coated with silicone grease or Vaseline or something like that. And it spins, and basically the pollen goes smacking into it. The big advantage of these is they're really not, since it just spins in a circle, it's really not sensitive to wind direction. The rotorod devices are really not sensitive to wind direction. The Burkhardt devices are always situated with a fin on them, so they always face the wind. But if the wind's blowing real hard, that means the motor speeds up a little bit and you actually collect a little more air. And if the wind is not blowing so hard, it actually slows down a little bit and you collect a little bit less air. And so it is a little sensitive to wind direction. These rotorod devices, basically they come with a fixed sample head, which is really sensitive to rainfall. If you run one of these during the rain, you wash off everything that's bound to that sample head, or a retracting sample head, whereby when it starts to spin, the rod comes out, and when it stops spinning, the rod goes back in. So this is a little less sensitive to rainfall, although if you've got a nice heavy rain going on, when you're actually sampling, it will wash off a lot of the pollen on the thing. You then take these little rods and you put them in a lucite adapter, and you put a stain on them, you put a cover slip over them, and you look at them in a microscope, and you count the number of pollen per area on the slide, and you know how much air these things have rotated through. And these rotorod has always got pi as a factor in its calculation, because it's spinning in a circle. But you spin how much, you calculate how much air these things have rotated through, and then you just divide the number of pollen for a day in the amount of air that's been collected for a day, and you get pollen per cubic meter of air. This is one that's actually mounted upside down, but it kind of shows this thing in action, and you can see them spinning. Normally these are turned over, so they're again trying to protect the rods from rainfall and getting an unusually low count, because you're getting a rain to wash off your rods. There was an article a few years ago, and this question pops up on board examinations frequently, comparing a rotorod sampler and a Burkhardt spore trap. Both instruments appear to record the same relative changes in particle concentration. The Burkhardt, however, is much superior for sampling particles lower than 10 microns. And so that's going to be mold spores, because most mold spores are smaller than 10 microns. So for the rotorod and the Burkhardt, they give approximately the same spore estimations for pollen. And so the National Allergy Bureau will accept pollen counts from a rotarod device, but they will not accept spore counts from a rotarod device. If you want to report spore counts to the National Allergy Bureau, you have to use a Burkhardt device or similar aerobiology spore trap. Now, this is another, the Allergenco is mostly for inside use, but we just set it up for outside use and demonstrated that at most wind speeds, like 90% of the time in Kansas City, the wind speed is not over 15 miles an hour. And so for the most part, the Allergenco produces very similar spore counts to the Burkhardt, and we published that information back in 2000. And basically the Burkhardt and Allergenco collectors displayed similar collection characteristics at all wind velocities for both pollen and spores. In other words, even if the wind was blowing pretty good, the counts were pretty similar because the inlet velocity of this Allergenco is pretty high. There were slight differences. The Burkhardt gave lower counts when absolute particle counts were low, and similar values when absolute particle counts were high. I think the Allergenco was a little better for the tiny spores. So all of these collectors have collection efficiencies. And when you get down to the 1 or 2 micron spores, the tiny little Aspergillus penicillin type spores, the ascospores and things like that, the collection efficiency starts to fall off pretty rapidly. And the collection efficiency, of course, is a function of the inlet velocity and the shape of the collecting head. And so the Allergenco had a really nice shaped collecting head. And this was bought up by a company, and they used it to produce a plastic model of the Allergenco collecting head that goes on a much more programmable type of vacuum pump here. And so you see these used a lot, mostly for indoor air sampling. And I think this is what Kevin's group is still using for indoor air sampling. But this is the comparison, and you can see they're pretty much the same. A lot of times, you get a little deviation, but they center around the same, and on average, they're pretty identical to use. Again, though, you've got to cover this Allergenco to keep rain from coming in the top. You have to think of these spore collectors as being outdoor devices, and so they're out in the weather and get a lot of rough use. Let's look at the pollens that we see in Kansas City, and this is kind of by appearance. Just check for a second. Okay, this is kind of by appearance. We generally see elm, maple, and cedar early in the season, depending upon how cold it is in January. We can see cedar in January, depending upon how cold it is. But typically, the first pollen we see, if it's a nice cold winter and spring, is the elm trees. And this is generally the end of February, 1st of March. And the elm pollen has a particular appearance. Now, the appearance that you see here is the pollen grain that has been collected on a greased slide, stained with a stain that makes it swell and that colors the various characteristics of the pollen grain. In this way, a human can look at these things and say, yep, that's an elm pollen under the microscope. There's some characteristics that can be described. It's stephanoporate. That basically means it's got pores that are around the outside edge here and none in the middle. They're 4 to 7, typically 5. The surface is fairly smooth, but the exine, the internal feature here, is fairly undulated. And of course, everybody knows what an elm tree looks like. Kansas City's got a lot of elm trees. They're mostly a paper type elm that were imported into the United States in the 30s and 40s from China and oriental elm trees. And they were planted because in the early part of the 20th century, Dutch elm disease came around, and it was a fungus that was carried by beetle, and the beetle would get in the tree and bore into the tree, and the fungus would get in the tree and kill an elm tree. And it's still around. It's still endemic in our area. And so every now and then I'll have an elm tree come up around here out in the pasture or out on the edge of the pasture. And it grows to maybe 10 feet tall, and then it dies because basically the borers got in it and the fungus got in it, and Dutch elm disease has killed the tree. So these elm trees around Kansas City were brought in to replace the beautiful American elms that Kansas City used to have. They're very prolific growers. You see them all over the place, and they make a great tree. They make a wonderful shade tree. Maple trees, we have many, this is the national tree of Canada, but we have many kinds of elm trees in our area, including sugar elms, although I don't know of anybody in the area who makes maple syrup. Maple pollen has a characteristic shape to it, and it is tricolporate or tricolpate, and that means basically it's got 3 slits in it, and this is one that you're looking at sort of head on, and you can see the slits in it. The egg sign is a little rough. It's a little hard to see from this this picture, but the maple tree is the one that produces the helicopters. This is the double-sided helicopter. The single-sided helicopter is produced by an ash tree. We don't have as many ash trees in our area, so ash is not in this list, but ash comes along about the same time as oak does. And of course, elm has a particularly shaped leaf, and it's one of the trees that you really should be able to identify when you go out walking or so. Cedar trees are basically juniper trees, and junipers and cedars and everything, including bald cypress, are in the Cupreaceae family. The pollen are basically completely round with no openings in it, but when I put stain on it, the stain causes the inside of the pollen grain to swell, and it breaks out of the husk that sort of surrounds the pollen grain. And so you get these Pac-Man-like structures on here. And so this is the outside of the pollen grain that broke open, and this is the actual pollen middle that came popping out of it. And this is a very characteristic of cedar or Cupreaceae. And you can see that I've got out by the barn. And if I take something, when this cedar tree has turned yellow, actually when it flowers, it turns brown. And then when it really starts to pollinate, it turns yellow. And if you throw something into this cedar tree when it turns yellow, it looks like a puff of yellow smoke coming out of there, and it really produces the pollen. Fortunately, or maybe unfortunately, these all choose about the same time to pollinate, to release pollen in the spring. So if the temperature is a little cool and we get a little rain, we're probably going to have a pollen release. And this cedar pollen just all releases at once. And so you get really high levels of it on a few days a year. Cedar is an invasive species for us. It's moving up here from North Texas and Oklahoma and the Edwards Plateau. And also we get in the wintertime, when the weather's just right down in Texas and you get cedar pollinating, we get cedar pollen blowing in here from Texas, and we can have fairly substantial levels of cedar pollen blowing in here from Texas, even during December and early January. And we have actually, Estelle Leviton actually did some work along with Jim Anderson. Jim collected cedar pollen in Ontario, Canada, and sent it back down to Estelle, who had it sequenced, and it's exactly the same pollen that was released in the Edwards Plateau about a day before. So it took it about a day to travel all the way up from Texas to Canada, where it was collected. Oak trees, there are 50 something different varieties of oak trees in Missouri. Oak tree again is a tricolporate, and sometimes it's very difficult to tell the difference between an oak pollen and a maple pollen. But they do generally appear at different times of the year. The maple is generally much earlier, the oak is generally much later. But they make beautiful trees, and their pollen is released from catkins. And a catkin is a flowering structure produced by a tree that basically involves a long stem-like flexible structure that has numerous flowers on it. And so the rule of thumb, and these things, of course, clog up gutters and you get all over your car and things like that. It's called a catkin. But the rule of thumb for pollen is that if the pollen comes from a flower or something that really has a nice smell to it, you don't get a lot of that pollen in the air. It's it's transported from one tree to another by insects. And so it is entomophlous or insect pollinated. Typically, these don't cause allergy problems. But if you have a tree where it produces tiny little flowers and you don't see it and you don't smell it, it's just tossing its pollen into the wind and it produces lots and lots of pollen. And these are anemophilous or airborne pollinated, and they do typically cause allergy problems. Mulberry, again, is a tree that we have a lot of around here, and they are diporate. You have two pores on a mulberry tree, and they're in the Morus family of the Moraceae. A very common tree around here is the hedge tree or Osage orange tree, and that is also in the Morus family. And so we typically see a lot of hedge pollen in our count that we report as Morus or mulberry around here. And of course, mulberry produces a nice, very seedy fruit, but it takes many, many years. This is a mulberry tree out in my pasture, and that tree has probably been there for 50 years. It grows slowly, and they typically don't get to be this size. Normally, a mulberry tree will be only 5 or 6 feet tall, but it still pollinates and still produces a lot of pollen. The first weed we generally see around here is sorrel or dock sorrel or sheep sorrel, and it's the Rumex family. It typically is tricolporate again, and but it has a very characteristic structure on the pore, and that's how you identify most of these, by the overall shape and the way it picks up stain and what the pore looks like. But you see this out in the fields around here, and some people eat this. You can have a sorrel salad if you really want to experience the taste of wild greens, and it's a strong taste, and so you don't see it on menus very often unless it's a really strange restaurant. But it's the first weed that we see around here, and we actually have a lot of sorrel in our area. Even in the downtown area, you'll find vacant lots with lots of sorrel plants growing in it. And then we typically see grass, and the grass is generally coming along in May here. And we start to get grass in May and then June. And all grass is part of the Gramineae or Poa family, and they all are nice and round and monoporate. They have a single pore on them. And so grasses are things like, this is Timothy grass, this is fescue grass, this is Johnson grass. Even corn produces a round pollen with a single pore in it. Corn produces a really big pollen. It's really heavy and doesn't blow very far. So it has to be a really windy day before you're ever going to pick up corn on a collector. But other grasses like Timothy and Johnson grass produce, and even the Bermuda grass and the Bahia grass, all the pollen looks exactly the same, even though the allergy picture of the Johnson grass and the Bermuda grass and the Bahia grass tends to be different. And then we get a lot of plantains around here. Plantain is that weed where, you mow your grass and the next day you go out there and there are these little spear-like things sticking up, starting to bloom. And this is plantain or plantago. It's periporate. In other words, it's nice and round and has numerous pores on it, but generally less than 15 pores. If you see something that looks like this and it's more than 15 pores, it's generally in the Amaranthaceae. But there are two varieties. This is the English plantain or Plantago lanceolata. This is the one that produces the little spear. And you can see it starts to flower from the bottom and just gradually flowers week after week until it comes all the way out the top. If you collect these spearheads after they've pollinated and produce seed, and you grind them up and you mix them up with a little lemon juice or something, you get mucilage or Metamucil. And so an interesting botanical note there. And then we have American plantain, which is very similar. And again, it starts to flower from the bottom and moves all the way up to the top, except this has a much broader leaf. And you see this growing almost everywhere. When you start to look for it, you see it growing in driveways, you see it growing in cracks in driveways, and even in roads and things like that. And then ragweed. We are one of the ragweed capitals of the world. There is a farm over around Sedalia that produces ragweed pollen on a commercial basis. And so we grow a lot of ragweed. It comes in two forms, the giant ragweed or the tall ragweed, which can grow 7-8 feet tall, and the short ragweed, which can still grow 5-6 feet tall. But typically the short ragweed can also grow about 2 inches tall. So it can actually grow and pollinate underneath the level that your lawnmower blade reaches. So you'll see this, you'll see ragweed around, especially the giant ragweed, lots. You see it around on the side of roads, especially where you have a creek or something. You see a lot of giant ragweed. And if you look closely at almost any lawn, you'll see a little short ragweed growing. And this is ambrosia. It's in the Compositae group. It's trichoporate, except it's generally identified by the spikes on it. The exine has a short spike, whereas most Compositaes have a longer spike. So this is called sub-echinate. And remember the echidna, if you played Sonic the Hedgehog, the echidna had a longer spike on him. And so this is a shorter spike, but it's the ragweed. And they just look irritating from just looking at them. Then the chenopod looks kind of like a golf ball. And anything or most things that are described as something weed are in the chenopod, and they've been reorganized. Now it's mostly the amaranthus group, and things like pigweed and lamb's quarters and goosefoot and dockweed and ironweed and a whole bunch of other things that are called weed are all in the chenopod or Chenopodiaceae group. And that's one of the problems you get when you try to tell people what they're actually allergic to. You might tell them they're allergic to pigweed, and what they have always thought was pigweed was not the same thing as you think is pigweed. But typically you're close because they're all in the Chenopodiaceae, Amaranthaceae group, and they all produce similar allergens, and their allergens have a lot of crossover in them. The other thing that's in this group that you see out recently is the kiwa. That is a seed grain that's very popular these days to be served as as a non-wheat, non-corn, non-POA grain that can be eaten with meals. It's grown a lot in South America and has been eaten as a seed grain for many years, but you see a lot more quinoa these days. I don't think you've seen a lot of allergy to quinoa out there. It's still not that common, and unless it's highly treated to remove the husk, it's pretty bitter. So those are top 10 pollens. Let's talk about the top 10 spores in our area. The number one is Cladosporum, and the spore cycle around here is when it's dry, spore levels go down, and mostly what you see is Cladosporum. Then it rains, the Cladosporum starts to grow and not necessarily produce so many spores that become airborne because it's nice, happy growing away. And so the Cladosporum level goes down when it's wet, but then when it starts to dry out, all this Cladosporum that's grown when it was wet now gets dry and now starts to become airborne and go floating around in the air. The opposite to that, the opposite to the Cladosporum, and the Cladosporum is generally characterized by the fact that you have one spore attached to as many as 3 other spore lines. In other words, this kind of grows as a chain, and then it starts to branch off in as many as 3 other lines. And so you have attachment sites on all of these spores. The other side of this is the ascospore. The Cladosporum was produced by an asexual reproduction. And so all the spores that were produced by the asexual reproduction look the same. They have the same genetics. The ascospore is many of the same organisms produced in a sexual stage. And so since you're mixing 2 sets of genes in the sexual stage, you get spores that don't always look the same, although you do have some characteristics about them. But ascospores basically have no attachment site on the end because they are produced as groups of 8 in a sac or ascus, which ascus is the old Latin word for sac. And they're produced about 8 at a time. The cycle is the ascospores are produced in this sac and it waits for wet weather. And when wet weather arrives and you get rain, a lot of times these spores are physically expelled from the sac and go out into the air. And so when we get rain, ascospore levels go up. When we get dry, ascospore levels go down. Cladosporum levels go up. When it gets wet again, Cladosporum levels go down and ascospore levels go up. And so lots of things can be called ascospores, and we generally refer to them as undifferentiated ascospores because we don't, even though some of them actually do have a name, we don't typically call them by name. Now, basidiospores are mushroom spores, and they are also a little bit connected to the wet cycle, but you see them delayed. In other words, if we have a lot of wet weather in about a week or so, we'll have a lot of basidiospores. And basidiospores are also, they are produced on a stool-like structure where you get 8 of them attached to the top of this structure. And so a basidiospore will have an attachment site and also a germ pore in here. And so they have a characteristic that they have the 2 sites, plus they're always asymmetric because they're produced 8 at a time, only they're produced around a central spike. And so there's plenty of room to grow on the outside of the basidiospore, but not a whole lot of room to grow on the inside of the basidiospore. So it's produced around this central spike. So they're always asymmetric. If you try to draw a line down it, you'll notice that one side is going to be always a little bigger than the other. Then Alternaria has a long history in allergy. This was the spore that was connected with allergy and asthma deaths by some researchers up in Minnesota. When Alternaria was high, they got more people in the emergency room. They got more people dying of asthma. It's interesting. It's kind of a club-like spore structure. It has an attachment site on both ends here because it kind of grows as a chain. It has longitudinal and transverse septations, and it's typically bigger, whereas a cladosporum might be 3 to 5 microns across, and Alternaria might be 10 to 15 microns. And so even though we give even a tiny little ascospore that's basically a couple of microns, it's barely big enough to see on a microscope, it counts as one spore in a pollen count. And Alternaria that's big enough so that at 1,000x it'll occupy almost half of the field sometimes in a microscope, it also gets the count of one airborne spore. And so you're breathing in, if you're breathing in an Alternaria, you're breathing in about 10 to 20 times more material than if you breathe in a little ascospore. But that's just the way we count them, and that's the way we've always counted them. Smuts are plant pathogens. Smuts and rusts are these days being grouped along with basidiospores a lot of times, but they're uretaspores, and they're smuts and rust. These are plant pathogens. You see them on crops growing around here. You see a lot of it in the air when crops are being harvested. We get corn smut, we get wheat smut. So if the corn is being harvested, all the smut that's grown on that becomes airborne as the harvester comes through and shakes everything up. Again, we get rusts around here. We get wheat rust and corn rust. And again, these become airborne when you see a lot of agricultural activity. Typically, you don't see these inside. Epicoccum, again, is a spore type that you see a lot of. These are, of course, produced asexually, and so they all look the same. They kind of have that soccer ball look to them with various septations throughout, and lots of septations. And again, these are big. These are as big as an Alternaria. Pithomyces, again, are pretty good size. They're characteristic, kind of a barrel shape with longitudinal and transverse septations, but a rounder or more oblong appearance. And again, attachment sites on the end. Helminthosporum, bipolaris, Dreschleria. These all look the same. It's a spore. It's got an actual lipid kind of vacuole in it. You can't tell the difference between the bipolaria, Dreschleria, and Helminthosporum until you see them sporulate. One sporulates out of the end, one sporulates out of the side, and one sporulates out of the middle. And so they just counted as a group. They just grouped together. And then Aspergillus and Penicillium, which are produce tiny little spores. This is the Aspergillus, and this structure here that you see in the middle of this group of Aspergillus spores is known as an aspergillum. And if you watch them in Pope or something like that, you'll see the guy who comes down the aisle with the holy water, sticks his aspergillum in there and sprinkles it to one side and then the other. And that's where the name comes from, Aspergillus, whereas Penicillium is produced on a finger-like structure and kind of uses the old Latin word for paintbrush or penicillius. And again, the spores for Aspergillus and the spores for Penicillium under the microscope, they look almost exactly the same. They're really small. You can barely see them. And so they get counted as Aspergillus Penicillium in one group. And that kind of brings us to the end of where I kind of normally stop this talk. You can also do sampling indoors for spores, but that's a talk for another time. So I thought we'd end right now and see if I have any questions.
All right, thanks, Charlie. Appreciate that. As always, it was fantastic. Any questions from the group today? quiet. To the fellows, I will tell you that your boards will have many questions related to information that was discussed today. Quite often they'll have pictures of a given mold and you'll go, oh, fantastic, I recognize that that's aspergillus, and then they'll ask some secondary questions. So anyway, good to keep up on this information, and I'm going to build that into more of our Jeopardy down the road, because I know that these questions are common. So anyway, well, Charlie, thanks again. We appreciate you being there and giving your free time. I hope retirement's been good to you, and hope we can count on you in the future again.
Oh yeah, we're enjoying it. We'll come back and talk about actually how to identify spores and show these pictures again, maybe.
That'd be fantastic. That'd be great.
Okay, thank you, Chris.
Okay, thank you. Thanks all.
Okay.
All right,
Bye-bye.
this is going to end COLA today, so we'll see everybody again on Friday. Thank you.